So in today's video, we're going to talk about ubiquitination and nitrogen degradation. On this first part of the video, we are going to talk about ubiquitination and its mechanism. You need to know that ubiquitin is a type of protein. It functions as a marker or a tag for other protein to proceed for protein degradation. Why? Because these proteins are toxic protein, misfolded protein, unused protein, and also worn out protein. This is the chemical structure of ubiquitin. For it to perform its function, ubiquitin needs to undergo 3 steps, which are activation steps, conjugation steps, and also ligase step. Tagging process starts with the activation of ubiquitin using enzyme 1. As any other activation, this is an energy-dependent steps. ATP is converted into AMP. Well, that is the chemical process. So basically, what we're going to see is E1 is attached to ubiquitin like this. Next is the conjugation steps using enzyme 2. This step is very simple. Basically, ubiquitin is transferred from E1 to E2. Lastly is the ligase step using enzyme 3. During these steps, the protein that need to be degraded will be attached to enzyme 3. And enzyme 3 will transfer the activated ubiquitin from enzyme 2 to the protein. This process is called tagging. This step will be repeated using different activated ubiquitin from different E2 enzyme. The protein can be tagged in many ways. It can either be monoubiquitinated, multi-ubiquitinated, or polyubiquitinated. The next steps involve proteasome. Proteasome is a protein complex which degrades unneeded or damaged protein via proteolysis. Ubiquitin taggings will undergo recognition via proteasome cap. It will force the damaged protein into the proteasome for proteolysis and detach ubiquitin for reuse. Through proteolysis, the damaged protein will be cut into smaller pieces and will be degraded. That's all for now. Thank you for watching.
Once proteins are broken down into individual amino acids can be built proteins or build nucleotide base. But if we have all the proteins that we want and all the nucleotide bases that our cells can actually use, what happens to any extra leftover amino acids? The excess amino acid must be broken down inside our liver. So let's take a look on how we actually break down amino acids. The catabolism of amino acids involves 3 stages. First, transamination, deamination, and urea cycle. Transamination is a reaction where amino acids meets alpha keto acids. Amino group must be removed as there are no nitrogenous compound in the energy transduction pathway. A can be any amino acids. Let's say, for example, A is a 3C amino acids, which is alanine. And B can be any keto acids. However, the most common keto acid in this reaction is 5-carbon keto acids, which is the alpha ketoglutarate. Now the functional group of A and B exchange with each other. Therefore, C become a 5-carbon amino acids, which is glutamate. And D becomes a 3-carbon keto acids, which is a pyruvate. So deamination is a reaction where the amino group is removed from the glutamate using glutamate dehydrogenase enzyme, which will produce alpha ketoglutarate and ammonia. So now the ammonia can be fed into the urea cycle.
The activity of urea cycle enzymes is elevated to meet the increased rate of protein catabolism. And I'm so sorry for my poor drawing skill. The major regulatory step is catalyzed by CPS1, which is known as carboxyl phosphate synthetase 1, where the positive effector is N-acetylglutamate, NAG.